Hello. The next video this week is about functional genomics, or starting to figure out what genes do. I will primarily rely on the example of CRISPR-Cas9. You might be thinking to yourself, haven't we been talking about what genes do for the last couple of weeks? But I will remind you that while we have been talking about associations between genomic variants and phenotypes of interest, an association is not the same as a causation. I also have been drilling it in your head, hopefully, that a statistical significance in an association is not the same as biological significance. You might be seeing something just due to chance, and that genomic region might not actually be involved in your phenotype of interest. And again, I was kind of saying this with the polygenic risk score, is that this risk is a summary of these associations. It's not an ultimate fate. So really, we've just been talking about associations and trends and populations. Now we want to ask, what really is it that genes do? How can I design an experiment to figure that out? The field of functional genomics is concerned with identifying the function or biological role of genes and non-genic sequences in the genome. Another key feature of functional genomics is that it uses high throughput or genome-wide methodologies. This makes it distinct from genetics, which might be asking what is the function of a gene on a more classical gene by gene approach. In functional genomics, we can scan the entire genome for clues about the function of genes. I do want to focus primarily on CRISPR-Cas9 in this video, but I want to take one quick moment to introduce at least a few other examples of functional genomics methods. One class of example looks for DNA-protein interactions. So you can imagine that in the cell, there are proteins that recognize specific DNA sequences, and they may have something to do with regulating gene expression of those genes or some other structural organization of the genome. So you can design a type of study that we'll talk about later in the semester that looks specifically for DNA sequences that are bound by proteins. Another type of functional genomics method that we've already talked about is RNA-seq. So in RNA-seq, you're using the level of gene expression as a predictor of the function of a particular gene. So let's say that I have two different types of cells, say a liver cell and a lung cell. They each have the same genome, but they will be expressing different amounts of some genes. Say some genes may be expressed a lot in the liver cell and very little in the lung cell. The differences in the level of expressions of those genes give us a hint to the function of the gene. Some of the most classical genetic experiments for determining gene function rely on a certain class of experiments. So, one of those is something that we would call a knockout experiment. And that is what happens when you have a total loss of function in your gene of interest. So, what phenotype do you observe when that gene is completely gone from your organism? Another type of experiment that you might be interested in is a knock-in type of experiment. In this case, you insert a new DNA sequence into your genome to see what the function is of that exogenous or new sequence in your organism. Here I'll try to show you that CRISPR-Cas9 can accomplish all of these objectives. I am going to be intentionally vague and not provide a lot of detail. There are so many great CRISPR-Cas9 videos. I'm going to post a couple links on Moodle if you'd like some supplementary information. Here, I'm just wanting you to understand the basics of how it's going to work. Here in blue, I've drawn double-stranded DNA. So this is in your cell of interest. You are going to add a couple of things to that cell in order to do a CRISPR-Cas9 mediated knockout. So one thing that you'll have to add is something called a single guide RNA or sgRNA that I am going to draw like this. The most important parts of this sgRNA are a region that has complementarity to your targeted DNA region of interest. That is, where do you want to cut the DNA? So that I've highlighted with the little lines. And then you also have a region of the guide RNA that is going to bind Cas9. So another thing that you have to add to the cell, if it's not already there, is the Cas9 protein itself, which I'll just draw as a big blob. Cas9 is the enzyme that binds the single guide RNA and does the actual cutting. So let's start our CRISPR experiment. So we have our targeted DNA and we add a single guide RNA that is going to specifically recognize a particular place in the genome. We're really hoping that there are no off-target effects. That is, if I design the single guide RNA with complementarity specifically to this region of the genome right here, we are really hoping that it doesn't also bind over here. That would be really bad. So we're hoping for a specific interaction between our genome and the guide RNA. And next, if there's not a Cas9 protein already expressed in our cell, then we have somehow managed to express Cas9 protein in our cell of interest. And that Cas9 protein is going to be recruited to the DNA by the single guide RNA, and Cas9 cuts DNA, and it specifically makes double-stranded cuts. I know that I usually draw DNA as a single line, but I drew it as two this time because I really wanted to be clear to highlight that it is a double-stranded cut that's being made. Because the next step that's going to happen is something that happens by the cellular processes that repair these double-stranded breaks. So the cell can stitch this back together so that we have an intact DNA. This is just natural cell repair. But when it does so, it often results in indels. Remember, that is an insertion or a deletion. When the cell sticks those two broken pieces of DNA back together, sometimes adds a base or reduces some. And remember, earlier in this semester, we talked about the consequences of insertions and deletions, particularly within a reading frame of a gene. That can lead to frameshift mutations, which result in early stop codons or nonsense mutations. And that's how we end up with a knockout. Because if you have an early stop codon in a gene, that is very likely to completely disrupt the function of the protein that results from that gene. CRISPR-Cas9 can also be used for knock-ins, and the initial steps are the same. You have your guide RNA that you've designed to target a particular region of the genome. You express Cas9 so that you can cut at that region of the genome, but you have one more thing, and that is your exogenous sequence, or we can think of as your new gene, or in the context of what we've been talking about in this class, is maybe a different variant of the gene that you would like to see expressed in your organism. And importantly, that new gene would have some homologous sequence on the ends that specifically matches where you have designed CRISPR-Cas9 to cut in your organism. So now, when the cell undergoes repair, it is hopefully not going to do the type of repair that results in indels, but instead it's going to do homology directed repair, or HDR, which is instead going to result in your sequence that you supplied being inserted into the region that was being repaired. So that now your new gene or exogenous gene or a different variant of the gene that you supplied as a template is now included stably in the genome of your organism and can be inherited through future divisions of this cell. So hopefully you can start to think now about how we could use tools like CRISPR-Cas9 to make a specific targeted change in DNA that we could then see maybe result in a phenotype change. And this is different than the associations between gene variants and phenotypes that we saw in large populations. Now we have a hypothesis that this gene or this sequence or this variant is associated with this phenotype. We can test it. We can manipulate it, probably not in humans, maybe in a cell line or in a model organism, and we can observe that phenotype. And now, when we do this, we've gone beyond the association, and we really have started to define the causal role for that gene in our biological system.